Welcome to the Deep Dive. Today we're plunging into something truly fundamental, something happening constantly within you, right now as you listen. Cell division. It's this invisible, incredible process that underpins everything from growth and healing to, well, the very continuation of life itself. Our deep dive today is all about the cell cycle and its regulation. We're going to unpack the intricate system that governs how cells grow and divide. And, the surprising ways this whole process is meticulously controlled. Our mission for you is to extract the most important nuggets of knowledge from our detailed research and notes, giving you a real shortcut to being well-informed about this foundational biological process. Get ready for some serious aha moments and truly fascinating details.
It's astonishing when you think about it, isn't it? Billions of microscopic operations happening with, breathtaking precision inside every living thing. And when you understand how this works, you really start to see the bigger picture of life itself.
Absolutely. Okay, let's unpack this rhythmic dance of life. At its most basic, what is the cell cycle and why is it so fundamental?
Well, think of the cell cycle as the complete life story of a cell, from its birth right through division into two daughter cells. For most of the complex cells in our bodies, what we call eukaryotic cells, this story unfolds in four distinct chapters: M, G1, S, and G2. M phase, that's where the dramatic division mitosis happens.
Yep.
But before that, you have interphase, which is really a period of growth and preparation. That includes G1, where the cell grows larger, S phase, where the cell meticulously copies its entire DNA blueprint.
Replication part.
Exactly, the replication part. And then G2, where it does a final check before dividing.
So it's not a one-size-fits-all process, right?
All right.
Our notes mentioned cells seem to have very diverse capabilities when it comes to division. Are all cells constantly doing this?
No, not at all. And this is where the cell's, you could say, life choices come in. Our sources highlight three main categories. First, you have highly specialized cells that have essentially retired from the division game. They're built for their specific job and don't replicate.
Like what kind of cell?
Think nerve cells, muscle cells, or your red blood cells.
Right, got it.
Then there are cells that normally don't divide, but can be induced to if needed. Think of your liver cells or lymphocytes.
Ah, so they can be called back into action.
Precisely.
Yeah.
If part of your liver is removed, the remaining cells can start dividing to regenerate it.
Wow.
And finally, we have the real workhorses, cells with high mitotic activity, like the stem cells in your adult tissues. They're constantly dividing to replace old or damaged cells, keeping you healthy.
That makes perfect sense. So constantly dividing. How long does a typical cell cycle even take? I imagine it varies wildly.
Oh, it does. It varies considerably, which is fascinating in itself. For a typical rapidly proliferating human cell, the entire cycle might take around 24 hours.
Okay, a full day.
Yeah, roughly. And within that, G1 can be about 11 hours, S phase maybe 8 hours, G2 around 4 hours, and the actual M phase, the division itself, is often just about 1 hour long. Pretty quick comparatively.
Just an hour for the main event.
Yeah, but then you have cells that are in a real race against time. Budding yeasts, for example, they can complete their entire cycle in only about 90 minutes.
90 minutes.
And even faster, sometimes 30 minutes or less, are the cells in an early embryo right after fertilization.
That's incredibly fast. And our sources mentioned something unique about those early embryonic cell cycles. What's going on there? Why the rush?
What's really remarkable is that these early embryonic cells prioritize rapid multiplication. They rapidly divide the cytoplasm of the egg into many, many smaller cells without the cells actually growing in size between divisions.
So they skip the growth phases.
They essentially skip G1 and G2 phases entirely. They just consist of straight S phases alternating directly with M phase. It's all about quickly increasing cell number by partitioning the existing egg volume, not about individual cell growth or elaborate preparation. It's like a quick way to build a lot of cells fast.
Okay, so if cells are making these life choices and going at different speeds, how do they know when to start this whole process? Sounds like there must be some crucial decision points guiding them.
Exactly. There are critical decision points, sort of like traffic lights for the cell. They're regulated by both signals from the environment and internal signals monitoring the cell's own status.
Tell me about those.
Okay, so in budding yeast, there's a major regulatory point called start. It's in late G1. Once a yeast cell passes start, it's fully committed. It will enter S phase and complete one division cycle.
No turning back.
Pretty much. And passage through start is tightly controlled by external signals, like are there enough nutrients available, and even the cell's own size. So for example, if nutrients are scarce, yeast will literally hit the brakes at start and enter a resting state. They won't commit if conditions aren't right.
Smart. And do our animal cells have something similar, a point of no return?
Yes, absolutely. The proliferation of most animal cells is similarly regulated in G1. We have a decision point in late G1 called the restriction point.
Okay, the restriction point.
It's analogous to start in yeast, but the key difference is how it's controlled. While yeast worry about nutrients, animal cells at this point are primarily listening for extracellular growth factors.
Ah, signals from outside telling them to divide.
Exactly. Without the right go signals from growth factors, they won't proceed past the restriction point.
Which leads perfectly into the concept of G0, right? The quiescent state. What happens if those growth factors just aren't there?
Well, if the appropriate growth factors aren't available in G1, the cell simply stops at that restriction point. It doesn't go forward. These arrested cells then enter a quiescent or resting stage called G0.
So they just wait.
They can wait for a long time, sometimes for your entire life without proliferating. They're still metabolically active, doing their specialized jobs, but they halt growth and significantly reduce protein synthesis.
Can you give an example?
Sure. A great real-world example is skin fibroblasts. They're typically arrested in G0, they just hang out. But if you get a cut, they're stimulated to divide for wound repair by platelet-derived growth factor, which gets released during clotting.
So it's like they're waiting for an emergency call to action.
That's a perfect way to put it.
Okay, so cells aren't just blindly moving forward. They have these pauses, these quality control mechanisms. Our sources call them cell cycle checkpoints. What's their role? Are they like tiny internal police officers?
That's a great analogy. Think of them as vigilant guardians or quality control inspectors. They absolutely allow things like DNA repair to happen, or they ensure processes are complete before the cell progresses.
So what are the key ones?
Well, the G1 checkpoint is crucial. It allows DNA repair before the cell even thinks about entering S phase. You really don't want to copy damaged DNA.
Right, that sounds disastrous.
It would be. So this is like a pre-flight safety check before DNA copying begins.
And what about during replication itself or after it's done?
Then you've got the S-phase checkpoint. This provides continual monitoring of DNA integrity during replication. It's like an ongoing diagnostic check.
Catching errors on the fly.
Exactly. Making sure incorrect bases are fixed or replication gets completed properly. Then, at the G2 checkpoint, the cell senses any unreplicated DNA. If the genome isn't fully copied, it halts the cycle, preventing entry into M phase.
Makes sense. Don't divide if the instruction manual isn't fully copied.
Precisely. And finally, there's the spindle assembly checkpoint. This is like a meticulous traffic controller during M phase itself. It monitors chromosome alignment on the mitotic spindle. It ensures that a complete set of chromosomes is distributed accurately and equally to the two new daughter cells. It's all about maintaining genetic integrity.
Wow, okay, this is all incredibly regulated. Now here's where it gets really fascinating. The discovery of the core molecular controllers driving this. Our sources mention these pioneering cell fusion experiments back in the 70s, Rao and Johnson.
Ah, yes. Their experiments with mammalian cells were revolutionary, absolutely groundbreaking. They essentially fused cells that were in different stages of the cell cycle. And what they found was amazing. The cytoplasm of a cell that was actively dividing a mitotic cell contains some kind of diffusible factors, things that could float around, that could actually induce mitosis in a cell that wasn't dividing.
So something in the dividing cell was triggering division in the other cell.
Exactly. It was clear evidence that there were active chemical substances driving cell cycle progression, not just passive timing.
And those substances led to identifying something called maturation promoting factor, or MPF.
Yeah.
What's that?
Right. MPF was found to be the key player that actually initiates entry into M phase, into mitosis. And what's really insightful is that MPF isn't just one molecule. It's a complex, a dynamic duo made of two subunits.
Okay.
One is a catalytic subunit. It has kinase activity. Think of it like a tiny molecular switch that adds phosphate tags to other proteins, changing their behavior.
Adding a tag to turn things on or off.
You got it. And the other subunit is regulatory, called cyclin. The name cyclin was coined because its concentration rises and falls in this predictable cyclical pattern with each cell cycle.
It cycles.
It cycles. It fluctuates precisely when needed, like a conductor appearing just for certain parts of the symphony.
So what does this discovery of MPF mean for the bigger picture of regulation? How do these molecular switches dictate the pace?
Well, this led to the understanding that these MPF-like enzymes are part of a whole family of universal controllers called cyclin-dependent kinases, or CDKs.
Ah, the famous
The
CDK.
very same. Researchers back in the 70s identified genes like CDC2 in fish and yeast and CDC28 in budding yeast, and their protein products turned out to be incredibly similar to MPF's catalytic kinase subunit.
So the yeast genes were coding for these CDKs.
Exactly. This was a monumental discovery. It established that one CDK, CDK1, is a universally conserved cell cycle regulator present in all eukaryotes, from yeast all the way to us humans.
The master conductor.
It's the master conductor for the whole cycle, yes. But crucially, it needs its cyclin partners to tell it when and what to conduct.
Okay, so it sounds like different cyclin CDK complexes might work like different keys for different locks throughout the cell cycle. Can you give us a feel for how they manage the transitions?
That's a perfect analogy. Think of the CDK as the engine, and the different cyclins as the specific ignition keys that turn it on for different stages or jobs. In yeast, for instance, CDK1 partners with specific B-type cyclins to drive the G2 to M transition, the big division push.
Right.
But to get past start, that first decision point back in G1, CDK1 pairs up with a completely different set of G1 cyclins.
Different key for a different door.
Exactly. And for DNA replication in S phase, it associates with yet another set of B-type cyclins. In mammalian cells, we see a very similar pattern. Cyclin B builds up and teams with CDK1 during G2, getting everything ready for M phase. It's a highly precise system of partnership for every stage.
Okay, so these CDK enzymes are clearly powerful. How does the cell make sure they're controlled so precisely? It can't just be an on-off switch based on the cyclin, can it? Our sources mention 4 key ways.
No, it's much more sophisticated than just on-off. There are at least 4 critical dials or mechanisms to fine-tune CDK activity. First, as we said, is simply the association with their cyclin partners. CDK activity is directly regulated by whether the right cyclin is present and at what level. No cyclin, no activity. That's fundamental.
Okay, step 1. Find your partner.
Step 1. Step 2 is activating phosphorylation. Even when a CDK has its cyclin partner bound, it usually needs another modification, a chemical phosphate tag added at a very specific spot by another enzyme called CK, CDK activating kinase.
So it needs a booster shot, basically.
Kind of like turning the ignition key fully to start the engine, yes.
Got it. Cyclin binding, activating phosphate. What else?
The third mechanism involves inhibitory phosphorylation. So imagine the cell also has a brake pedal system. Certain enzymes, like one called WEE1, can add inhibitory phosphate tags, usually near the CDK's front end. These act like brakes, shutting down CDK activity even if a cyclin is bound and the activating phosphate is there.
So you can actively put the brakes on.
You can. And the fourth mechanism involves proteins called CDK inhibitors, or CKIs. These are specialized proteins that directly bind to the CDK or the CDK cyclin complex and physically block its activity.
Like a roadblock.
Exactly like a roadblock. In mammalian cells, we have 2 main families of these CKIs. One family, the INK4 inhibitors, are sort of G1 specialists, inhibiting the CDKs that drive early progression. The other family, CYP-KIP inhibitors, are broader acting. They can inhibit CDKs involved in multiple phases.
And yeast have them too.
Oh, yes. Budding yeast has a key CKI called SYK1. It's particularly important in G1, and its degradation is actually the crucial signal that allows the cyclin CDK complexes to finally initiate DNA replication. So it's a complex dance: accelerators, brakes, and roadblocks.
Okay, here's where it gets really impactful, connecting these, incredibly intricate cellular mechanisms to a critical human health issue: cancer. Let's talk about cyclin D1 and growth factors. How do they fit into this control system?
This is a really crucial link. Cyclin D1 is one of those cyclins active in G1. Its production, its synthesis, is directly triggered by those external growth factors we mentioned earlier, the signals telling the cell to grow and divide.
Okay.
Its synthesis continues only as long as those growth factors are present. If they disappear, cyclin D1 is very rapidly degraded. It's got a short fuse.
So it's tightly linked to the growth signal.
Very tightly. So as long as the growth signals are active, cyclin D1 builds up, forms complexes with its CDK partners, CDK4 and CDK6. And these CDK4006 cyclin D1 complexes are what actually push cells past the restriction point, that major G1 decision point.
Right, the point of no return for division. So what does this mean for cancer? What happens when this finely tuned system goes wrong?
Well, think about it. If you have unregulated, continuous production of cyclin D1, maybe due to a mutation, so the growth signal is effectively stuck on us, then the cell keeps getting pushed past the restriction point, even when it shouldn't. And that unregulated progression directly contributes to various human cancers. It's frequently overexpressed in lymphomas, breast cancers, and others.
Wow.
And similarly, mutations that inactivate those CDK inhibitors we just talked about, the INK4 family that normally acts as a brake on CDK46, those inactivating mutations are also very commonly found in human cancer cells.
So losing the brakes or having the accelerator step down.
Exactly. It shows how disrupting even one piece of this finely tuned regulatory machine can have profound, really dangerous consequences, leading to uncontrolled proliferation.
And a major player in this cancer connection, a classic example everyone hears about, is the RB protein, right? Retinoblastoma protein. It's a critical tumor suppressor gene. How does it act as a guardrail?
Yes, the RBE2F pathway is absolutely central to understanding this link between cell cycle control and cancer. RB is a fascinating protein. In its active underphosphorylated form, which is what you find in resting cells or cells early in G1, RB literally clamps down on a family of transcription factors called E2F.
Okay, what do E2F factors do?
E2F factors are the guys that turn on a whole suite of genes needed for cell cycle progression, particularly for entering S phase. Things like cyclin E, enzymes for DNA synthesis, and so on.
So RB stops E2F from turning on the Go genes.
Precisely. When RB is bound to E2F, it acts as a repressor. It physically blocks E2F from activating transcription, effectively putting the brakes on those cell division genes.
But then something must release the brakes.
Exactly.
Right.
And here's the key link back to cyclin D. When RB gets phosphorylated, gets those phosphate tags added specifically by those CDK 4006 cyclin D complexes.
The ones activated by growth factors.
The very ones. That phosphorylation causes RB to change shape and release E2F. Now, E2F is free, and it can go and activate transcription of its target genes, like cyclin E, pushing the cell cycle forward towards S phase.
Okay, so that explains how cyclin D helps release the RB brake.
Now, how does CDK2 cyclin E then help orchestrate the final push into S phase, into DNA replication itself?
Right. CDK2 cyclin E is the next key player. Its activity is sort of the trigger for S phase,
Okay.
but it's also tightly controlled. Initially, in resting cells or early G1, CDK2 cyclin E activity is held in check by one of those CDK inhibitors we mentioned, a CYP protein called P27. So P27 is another brake.
Okay, another break.
But the growth factor signals that activated cyclin D production also start to work against P27, often reducing its levels. Then, as the cell passes the restriction point, more cyclin E is produced thanks to that E2F being released by RB phosphorylation.
Now, here's a clever bit. The rising levels of cyclin D complexes, CDK 4006 cyclin D, actually help activate CDK2 cyclin E indirectly. They start binding up P27, effectively sequestering it, pulling it away from the CDK2 cyclin E complexes.
Distracting the inhibitor.
Kind of, yeah. So with less P27 bound, CDK2 cyclin E starts to become active. Then active CDK2 actually phosphorylates P27 itself, marking P27 for complete destruction via the cell's disposal system.
So it removes its own inhibitor.
It helps remove its own inhibitor. Finally, with the P27 brake fully released and CDK2 cyclin E activity ramping up, these complexes can trigger the initiation of S phase. They do this by activating key proteins, like the MCM helicase proteins, at the DNA replication origins. That's the final start DNA copying now signal.
Wow, that's a lot of intricate steps just to get DNA replication started. Which, raises a really important question. How does the cell make absolutely sure that replication starts at thousands of different origin points across its huge genome only once per cell cycle? Getting that wrong sounds like chaos.
It absolutely would be chaos. And the cell has a beautifully elegant solution for this, often called the licensing mechanism.
Licensing, like getting a permit.
Exactly like getting a permit, or maybe like checking out a book from a library once. Replication initiation is carefully separated into two distinct steps. First, early in the cell cycle, probably in G1, specific locations on the DNA, the origins of replication get licensed. This means they're approved for replication. A special protein complex, the licensing factor, attaches to each origin.
Okay, they get stamped for approval.
You could think of it that way. Second, later on, when S phase actually begins, the replication machinery only initiates copying at these licensed origins. It ignores unlicensed ones. And here's the crucial part. As the replication forks move away from the origin and copy the DNA, that licensing factor is removed or inactivated. This leaves the origin unlicensed.
So it can't be used again right away.
Exactly. It cannot be relicensed and therefore cannot be copied again until after the cell has fully divided and re-entered G1 of the next cycle. It ensures everything is copied once and only once.
So what is this magical licensing factor? What's the protein doing the stamping?
In eukaryotes, like our cells, The core licensing factor is a protein complex called MCM, which stands for mini chromosome maintenance. It's actually a complex containing DNA helicase activity, the enzyme needed to unwind the DNA double helix.
Okay, the unwinder.
Yes, MCM must bind to the DNA at the origin for replication to initiate there. And crucially, after replication has begun at an origin, another protein called geminin steps in. Geminin acts like a temporary blocker. It binds to components needed for MCM loading, effectively preventing MCM from binding again to that same origin during S phase or G2.
Gemin prevents relicensing.
Precisely. Then, at the very end of mitosis, geminin is purposefully targeted for degradation. Its removal allows MCM to once again bind to the DNA origins as the cell enters the next G1, relicensing them for the upcoming S phase. It's a really neat cycle of licensing, blocking, and relicensing.
That is a clever system. Okay, but what happens when things go wrong with the DNA itself? Let's say it gets damaged by radiation or chemicals. How does the cell respond then? It must have an emergency brake system for DNA damage.
You're absolutely right. There are critical emergency brakes specifically for DNA damage, the DNA damage checkpoints.
Yeah.
Cell cycle arrest at these checkpoints is triggered primarily by two master sensor protein kinases, ATM and ATR.
ATM and ATR, what do they sense?
They are like the cell's first responders to DNA damage. ATM is mainly activated by severe damage like double-strand breaks in the DNA helix. ATR, on the other hand, is activated by less catastrophic damage like single-stranded DNA breaks or stretches of unreplicated DNA, which can happen if replication stalls.
Okay, so they detect different kinds of trouble. What do they do once they're activated?
Once activated, these sensors act like commanders. They phosphorylate and thereby activate a pair of downstream checkpoint kinases called CHK2, activated mainly by ATM, and CHK1, activated mainly by ATR.
So ATM, the tivantin, CHK1, CHK2, then what? How do they stop the cycle?
CHK1 and CHK2 are the enforcers. Their main job is to target and inhibit a set of critical go-ahead proteins called CDC25 phosphatases.
CDC25, what do they do normally?
Remember how CDTX often need activating phosphorylation but also have inhibitory phosphorylations put on by WEE1?
Yeah, the brakes?
Well, CDC25 phosphatases are the ones that remove those inhibitory phosphate brakes from the CDKs, allowing them to become fully active.
Ah, so CDC25 takes the brakes off CDKs.
Exactly. So by phosphorylating and inhibiting CDC25, CHK1 and CHK2 effectively keep those inhibitory brakes on the CDKs, halting cell cycle progression.
Clever. Does it work the same way in all phases?
It targets the relevant CDC25s for the specific phase. For example, at the G1 and S phase checkpoints, CHK1 and CHK2 phosphorylate CDC25A, which normally activates CDK2 complexes needed for S phase entry and progression. This leads to CDC25A's rapid degradation and CDK2 inhibition, arresting the cell right there. At the G2 checkpoint, CHK1 and CHK2 phosphorylate and inhibit CDC25C, which is the phosphatase needed to remove inhibitory phosphates from CDK1 cyclin B. Inhibiting CDC25C blocks the activation of CDK1, preventing the cell from entering mitosis and arresting it in G2. It buys precious time for DNA repair.
It's a really precise chain of command to hit the pause button exactly where needed. Now, there's another key player, especially in mammalian G1 arrests, isn't there? The famous P53, often called the guardian of the genome.
Yes, absolutely. P53 is incredibly important, particularly for the G1 DNA damage checkpoint in mammalian cells. When ATM and CHK2 detect DNA damage, they phosphorylate P53. Normally, P53 is very unstable and gets rapidly degraded, but this phosphorylation stabilizes it.
Makes it stick around longer.
Exactly. So DNA damage leads to a rapid surge in P53 protein levels, and P53 is a transcription factor. It turns on specific genes.
What genes does it turn on?
One of the most critical genes it activates produces a CDK inhibitor protein called P21. P21 belongs to that CYP family we discussed earlier.
The broad CDK inhibitors.
Right. This induced P21 protein then directly binds to and inhibits the CDK2 cyclin E complexes.
The ones needed to get into S phase.
Precisely. So inhibiting CDK2 cyclin E causes the cell cycle to arrest in G1. This gives the cell a crucial window to repair the DNA damage before it attempts replication. And if the damage is too severe to repair, P53 can actually trigger programmed cell death, apoptosis, preventing the propagation of a dangerously mutated cell. It truly earns its guardian title.
Okay, phew. That's a lot of breaking and checking. Now, let's finally turn to the main event itself, the dramatic transformations that occur during M phase, mitosis. You said this is all orchestrated by the master conductor, CDK1 cyclin B. It sounds like a grand choreography.
It absolutely is. A stunning, highly choreographed spectacle within the cell. CDK1 cyclin B phosphorylation triggers a cascade of events. One of the first and most visible is chromatin condensation. The DNA, which is usually long and sprawling, needs to be packaged up tightly.
Right, into those X-shaped chromosomes we see in pictures.
Exactly. This is driven by protein complexes called condensins. Think of them as molecular winches that coil and compact the DNA strands. These condensins are switched on by CDK1 cyclin B phosphorylation. As they work, they largely replace other protein complexes called cohesins, which were acting like glue, holding the two replicated sister chromatids together along their entire length since S phase.
So condensins compact and cohesins held them together.
Right. Condensation mostly replaces the cohesin linkage, except right at the central region, the centromere. This leaves the sister chromatids tightly linked only at the centromere, but highly condensed everywhere else, resulting in those characteristic compact mitotic chromosomes.
Makes sense. What else happens? What about the nuclear envelope, the barrier around the DNA? That seems like a major obstacle if you want to separate chromosomes to opposite ends of the cell.
It is, and it gets dismantled. The breakdown of the nuclear envelope is another major M-phase event orchestrated by CDK1. This involves several things happening simultaneously. The nuclear membranes fragment into small vesicles, the nuclear pore complexes that control traffic in and out just to dissociate, and the nuclear lamina, the protein meshwork supporting the nucleus depolymerizes, it falls apart.
How does CDK1 do all that?
It directly phosphorylates the key structural proteins involved, the nuclear lamins, internuclear membrane proteins, components of the nuclear pore complex. These phosphorylations cause them to disassemble precisely when needed.
Wow. And other organelles. Our notes mentioned the Golgi apparatus.
Yes, the Golgi apparatus, which is involved in processing and packaging proteins, also needs to be dealt with. It fragments into hundreds of small vesicles during mitosis. Again, CDK1 is involved, phosphorylating Golgi matrix proteins. This seems to inhibit the normal fusion of vesicles, causing the structure to break down so it can be easily and evenly distributed to the two daughter cells later on.
Okay. Nucleus dismantled, Golgi fragmented. What about building the machinery to actually pull the chromosomes apart, the spindle?
Right, spindle formation. This is absolutely critical. Early in M phase, CDK1 activation triggers the separation of the duplicated centrosomes. These are the main microtubule organizing centers of the cell.
They move to opposite sides.
They migrate to opposite poles of the nucleus. As they move, they mature, recruiting more proteins, like gamma-tubulin, which helps them nucleate microtubules much more effectively. At the same time, the dynamics of the microtubules in the cell change drastically. The relatively stable interphase microtubule network breaks down. It's replaced by a huge number of short, highly dynamic microtubules radiating out from each centrosome, forming the mitotic spindle.
Like building a whole new railway system.
It's a perfect analogy. Then, once the nuclear envelope breaks down, this new railway system can access the chromosomes. Spindle microtubules attach to the chromosomes at specific structures on the centromeres called kinetochores.
The connection points.
Exactly. This attachment initiates the complex pushing and pulling movements that eventually align all the chromosomes perfectly at the center of the spindle, forming the metaphase plate. They're all lined up, ready for separation.
This brings us right to the climax. The metaphase to anaphase transition. This is the moment where the sister chromatids finally separate. You said it's meticulously controlled, feels like the grand finale.
It truly is the climax, and the control is exquisite. Remember that spindle assembly checkpoint, the SAC?
Yeah, the one checking chromosome alignment.
That's the one. It's absolutely essential here. A complex of proteins, including the MAD and BOP proteins, acts like that meticulous traffic cop. It constantly monitors the attachment and tension on every single kinetochore. It sends a powerful wait signal, preventing the cell from proceeding to anaphase until every last chromosome is perfectly aligned at the metaphase plate and correctly attached to spindle microtubules from opposite poles. No chromosome left behind.
Okay, so once the SAC is satisfied, every chromosome is lined up perfectly.
Yeah.
What's the actual trigger? What gives the final green light for anaphase, for the separation?
That green light is given by the activation of a powerful enzyme complex called the anaphase promoting complex, usually abbreviated as APC, or sometimes APCC.
APC, what does it do?
The APC is an E3 ubiquitin ligase. Remember ubiquitin, the tag for protein destruction.
Right, the molecular tag for disposal.
Exactly. APC's job is to put that ubiquitin tag on specific key regulatory proteins, marking them for destruction by the proteasome, the cell's recycling machinery. Now, APC activation is actually initiated early in mitosis, but it's kept inhibited by the spindle assembly checkpoint as long as there are any unattached or improperly aligned chromosomes.
How does the checkpoint inhibit APC?
Those MADBUB proteins, when they assemble at an unattached kinetochore, they bind to and inhibit a crucial activating subunit of the APC called CDC20. So, unattached kinetochores generate a wait CDC20 signal, keeping APC switched off. Once all chromosomes are properly attached and aligned, the MADBUB complex disassembles from the kinetochores, the inhibition on CDC20 is relieved, and APC finally becomes fully active.
Okay, so APC is activated. What exactly does it target for destruction to trigger the big separation?
Activated APC, working with CDC20, APC-CDC20, has a very specific and critical initial target, an anaphase inhibitor protein called securin.
Securin, aptly named.
Very aptly named because it literally secures the link between sister chromatids. It does this by inhibiting another protein, a protease, a molecular scissor, called separase.
So securin inhibits separase.
Correct. So when APC gets activated, it ubiquitinates securin, marking it for destruction.
Destroy securin.
And when securin is destroyed, separase is released and becomes active.
The scissors are free.
The scissors are free. Active separase then goes and cleaves a specific subunit of that cohesin complex, the glue that's still holding sister chromatids together at the centromere.
It cuts the glue.
It cuts the last bit of glue. This cleavage breaks the link between sister chromatids, triggering their sudden and dramatic separation. That's the onset of anaphase. They start moving rapidly towards opposite spindle poles. But APC isn't done yet. It has another crucial role, especially towards the end of mitosis. As CDK1 activity naturally starts to drop, APC associates with a different adapter protein, CDH1, APC-CDH.
A second job for APC.
A second, crucial job. APC-CDH1 is responsible for completing the ubiquitination and destruction of the mitotic cyclins, primarily cyclin B. APC-CDC20 actually starts this process, but APC-CDH1 finishes it off efficiently.
Destroying the cyclin B that partners with CDK1.
Exactly. The complete destruction of cyclin B causes a sharp, precipitous drop in the activity of the main mitaric CDK, CDK1. And this drop in CDK1 activity is the master signal that drives the cell out of mitosis. It allows things like chromosome decondensation, nuclear envelope reformation, cytokinesis, the actual splitting of the cell to occur, and resets the cell back into G1 for the next cycle. It's the ultimate reset button.
Wow. What an incredible deep dive that was. We've just gone through the truly, mind-boggling precision and the multiple layers of regulation that govern something as fundamental as cell division. You've really seen how cells, the basic units of life, manage their own reproduction with such meticulous control, from the basic growth phases to these complex checkpoints and this whole army of molecular machinery. It's just amazing to think that all of this is happening trillions of times over inside you right now.
It really is a system of astounding complexity and, frankly, elegance. And it truly underlines how essential this tight regulation is for healthy cell function, for life itself. We saw the profound implications when these processes go awry, like in cancer. It just highlights the constant balancing act inside our cells. And it definitely raises an important question, doesn't it? What other fundamental biological processes, things we take for granted within our bodies, are governed by such intricate, finely tuned systems, just performing perfectly hour after hour, day after day?
That is a fantastic thought to leave you with. We definitely encourage you to keep exploring your curiosity about this incredible microscopic world that so fundamentally shapes our macroscopic lives. Until next time on The Deep Dive.